Thank you very much, and thank you for inviting me to speak today. I'm just going to try and share my screen. If you can tell me when that's come through, that would be great.
Yeah, that's come through.
Excellent job. Okay, well, as you can guess from the introduction, I'm the tree health officer covering the southeastern London. Now, my background, I've been involved with tree health and tree pests and diseases for almost 20 years now. Started off down in the southwest of England, but more recently I've been based up in Yorkshire and the northeast covering this role, but up there. So we're going to run through a few pests, well, one pest and some diseases today in relation to sweet chestnut. So we're going to have a look at Phytophthora remorum. We're going to have a look at cinnamomi. We'll have a look at sweet chestnut blight, which is Phytophthoras parasitica, and then also oriental chestnut gall wasp. So what I thought I'd do to start with is looking at sort of trying to understand what Phytophthoras are with regards to sort of phylogenealogy and looking at how they fit in compared to fungi. So you'll be able to see on the screen in front of you, you've got fungi sat over here. And then you've got, working your way across, you've got plants, and over in the heterocont here, you've got Phytophthoras. So they're very different and distinct as far as evolution goes from fungi. They're not fungi at all, they're water molds. And that will become a little bit more obvious and apparent as the types of environments we look at them in a minute. So splitting down Remorum into clades, into different groups again, we'll be looking at, to start with, Phytophthora remorum, which sits in clade 8. There's a few others which some of you may be familiar with in that same group as well, like Phytophthora lateralis and Ostracedra as well. So there's a number of rather effective plant pathogens that sit within that clade, within that group. So thinking about Remorum now and what it actually looks like, what it is. So we have a couple of structures that are of interest. First of all, we have the sporangia. So the sporangia contain zoospores. So those are quite highly motile little things. But these structures, these sporangia and the zoospores, which you can just see come up now, they're rather ephemeral. They don't last for very long in environments. They're rather they're not very tough, basically. They're open to attack from all sorts of other bits and pieces. But the zoospores have these flagellum tails, so they're highly motile. They can swim through water and mist as well, if there's enough mist in the air, which is why we're sort of starting to think about aerial transmission, which we'll pick up on a bit. But they're able to move around rather effectively. We then have chlamydospores. So these are the resting structures. They have a thick cell wall. They're certainly able to persist in the soil for at least 10 years that we know of and remain viable, and then go on to infect host species that they come into contact with again. So that's their survival strategy in effect. This is their short-term spread, getting around quickly, and this is their survival in the environment when there's no host material nearby. What do they actually look like in relation to sort of thinking about now? This is a leaf disc of Californian bay laurel, so not a host we tend to find in GB. But to give you an idea of scale, these are the little sporangia, and within each sporangia there are about 30, with Remorum, there are about 30 zoospores within each of those sporangia. So there's a number of spores all contained within those there. So it's very effective at reproducing itself, and it's rather a small little organism, especially when you look at it in relation to the scale of that leaf. And here you can just about, if you squint faintly, you can see some swimming zoospores as well. So highly motile is one of the things to sort of remember with Remorum. So now thinking a little bit more about life cycles. So Remorum it reproduces asexually, so in effect it clones itself. So we don't have any heterothallic reproduction, it's all homothallic. So you have the mycelium, I've shown you the sporangia and the zoospores and the chlamydospores. Those are all able to either, with the sporangia, release the zoospores, which then go on to germinate and re-enter material through hyphal penetration, or also with the sporangia they're able to directly germinate into their host. And chlamydospores, same thing, they can either produce the sporangia, which then go that loop again, and they're also able to directly, through hyphal penetration, infect a host. So what does that look like, taken rather sort of clinically in that diagram, actually putting it into some sense of what it looks like with a host? So you have the sporangia that appear on the host, usually, well, we're going to take this instance, sweet chestnut, on the leaf. They break through, produce those spores through the sporangium and then the spores, and then also through the direct germination or indirect through the chlamydospores as well. They go through some form of aerial transmission, sometimes soil with Remorum as well. Those zoospores land on a suitable host. In this instance, we'll say sweet chestnut leaf blade or some of the thin bark as well sometimes. Penetrates down through into the plant material and then starts the whole infection cycle off again. So what does that actually look like in relation to symptoms expressed on host? So these are a few sort of foliar samples we're looking at with Remorum and sweet chestnut. It's quite, the lesion edges are rather indistinct, as you can see on the left-hand side here. They're very watery. It looks a little bit like somebody described it to me once as ink on blotting paper, which I think is rather a good description. Those leaves, once they've once the disease has been through there, those leaves remain pliable, a little bit like wax-proof paper as well for a period of time. So although they'll look dead, they're still quite pliable rather than crumbly and friable like you would expect if for instance, they die from something else. So that's another sort of interesting diagnostic feature. Also thinking about sort of water and water availability and splash transmission and droplet transmission. If you think about water collecting on the leaf tips here and working its way back through, so you quite often find them, the starts of those lesions at the leaf tips. Or conversely, as you can see in this middle picture, sometimes they start from the petiole and work down the midrib. So they can come in from either direction, sometimes on the same leaf, as you can see within this one. It started at the top, might have even been a bit of sort of through into the middle of that vein there as well, but also starting from where the moisture was collecting down the bottom. So thinking about that transmission and water droplets is really important when you're looking for symptoms. What that then does is progress through the petiole into the woodier part of that new growth. So here you can start to see you've got necrosis of the underbark material here, and that starts to work its way down through as the symptoms progress, and it builds up a little bit of steam. In effect, it's sort of starting to work its way through. It then starts to look something like this. So in the earlier stages on the epicormic stuff, if that's where the infection started, you tend to get this foliar collapse. So you'll see odd bits of dieback that initially from a distance you can't explain as to why. As that progresses, that symptom expression becomes more and more extreme, and it will start to work its way through into sometimes the larger branch material as well. Or conversely, sometimes it can stay just as an epicormic infection on the lower part of the tree, and you'll just see this sort of repeated cycle of defoliation, regrowth of epicormic growth and defoliation again. So it can be quite cyclical sometimes in the right site environment as well. So humidity humidity does play an important role in how this disease cycles within the stand as well. If it can get up enough sort of a spore pressure as well, you'll then start to see some collapse within the crown as well. And sorry, this is a bit of a washed out photo, but what you can see is the foliage is starting to collapse in the upper crown and work its way down through. So this was a valley environment in the southwest of England, high levels of humidity, high spore pressure. There was a lot of larch on this site as well. So there was a vast amount of spore pressure raining down on the upper part of that canopy, which started to cause that defoliation. Same valley again down in the southwest of England. So this, to try and sort of contextualize, there was a significant amount of larch either side of these trees and actually throughout the whole forest block, with a significant Remorum infection in there. And here you can see it's really started to work its way down through the main stem of the tree. We're just almost left with epicormic regrowth now. So really under sort of a lot of stress and not doing very well. And ultimately, it can end up looking like this. It can end up in mortality of the tree as well. So it's rather effective pathogen in the right climatic conditions, I would say. And again, these are rather sort of extreme pictures, but what show you what can happen. Again, down in the southwest of England, just restricted to a few epicormic shoots on either side of these trees. So, not doing very well at all. Now, I know today is very much a sweet chestnut focus, but it's very difficult to take Remorum without talking about symptoms and the effect that rhododendron has on the transmission cycle as well. So thinking about points of infection within a woodland and how that and initial transmission into a woodland and how that might start. If you're near other points of infection, then it could well be an aerial, that it's come through aerial transmission, but longer dispersal. You know, we're talking of hundreds of tens or hundreds of miles here. You're looking at human interaction that's played potentially a significant part in that transmission. So be that through movement of machinery with soil, organic debris still attached to it, or plants for planting, or all sorts of bits. Plants for planting are a little less likely for Remorum in a woodland environment anyway, but it's rhododendron that acts as the initial catalyst in an infection quite often within woodland. So if you think about that soil transmission, it's easy to get a little bit of soil onto some rhododendron leaves, or a little bit of rain splash. So that's where you start to see the symptoms primarily in the woodland. It's going to be on the rhododendron first. So you'll get that foliar collapse to begin with. And again, you can see in the middle one here, we're starting to see a sort of similar running theme of watery lesions, watery necrotic lesions that start from points where moisture will collect. So again, thinking here of splash and sort of water splashes and things, and where it would collect, starts to work its way down the midrib. And here you can see just a bit closer in, so you can start to see sort of probably collected here and worked its way down each of these midribs. But again, can start and come in from the leaf tips as well. And as that progresses, you start to see a decline of that whole bush, and then you start to see the sort of old dieback where it's all gone through, retained foliage. Again, that retained leaves, they're very malleable still. They're not friable. They still feel like wax-proof paper. So like the sweet chestnut leaves I was saying about, they don't crumble in your hands as you would expect a leaf looking like that to do normally. And here you can see on the edges of the picture, you can still see that sort of infection going on and starting afresh in these newer bits of growth on the edges. So really important to think about Remorum of not only looking at the trees within the woodland, but also thinking about looking at rhododendron as well, because that's where it's likely to see where you express symptoms first that we're going to pick up on anyway. So thinking about Remorum with regards to how we control that. So it is a quarantine organism. It's subject to statutory plant health notices. Those are notices for control and destruction. There are also biosecurity measures associated with that as well. It's also a reportable disease as well, as you can imagine, as it's a quarantine organism. So if you suspect you're seeing Remorum on sites, then you need to report that, and ideally through tree alert. So the map you can see on the side here was our aerial survey activity last year. Primarily, sweet chestnut is a little bit more difficult to spot those fine symptoms, as we've seen earlier on looking at the epicormic infections. It's a little bit difficult to see that from the air. We can pick up to a certain extent the larger infections and the dieback that happens within the crown. So, but primarily we're looking at larch when we're doing these surveys. Issues last year, same as everybody, we had COVID restrictions, so we're now down to a single crew member in the aircraft, which has caused, it's still made it an effective survey, but it's taking more time because we've only got one set of eyes instead of three set of eyes. What we've also observed last year was a significant increase in the number of positive larch sites. That's primarily due to weather in the previous two autumns. Those of you that are coming along tomorrow, I'll discuss that a little bit further and looking at sort of weather patterns and the effect that that's had. But, you know, given that there's more spore pressure around within those larch sites, is there going to be more sweet chestnut sites? Possibly. It's always a possibility because there'll be a higher spore pressure. So that's Remorum over through. We'll have a look at cinnamomi now. So this is something that causes ink disease. So sits within a different clade now. So we were looking at clade 8 for Remorum. We're now into clade 7 for cinnamomi. There's another rather effective plant pathogen in that clade as well, Cambivera, for some of you that's come across that as well. That's really quite effective at killing beech trees in fairly short order. But cinnamomi has a huge host list. I think it's somewhere about in order of sort of about 400 different species for hosting. Causes all sorts of issues in other countries as well. Jarrah dieback, if anybody's heard of that in Western Australia, also has a real problem for avocado farming as well, causes avocado root rot. So, you know, not only is it an effective tree pathogen, it also has a significant impact on agriculture as well. So looking at the types of structures, you'll see a difference here. So we'll have the sporangia, which we saw before with Remorum, which contains zoospores. We've also got oospores this time, so letter E and F. Those two slides there are oospores, so that's where there's been sexual reproduction. And then again, you'll get asexual reproduction, the chlamydospores G through to K. Quite morphologically diverse of chlamydospores within Synonyms. So having a look, sort of as I was just saying there about heterothallic reproduction, so you need to have the two mating types available for that to occur, two compatible mating types. And those oospores behave in a similar way as chlamydospores in the fact that they can produce sporangium, and they can also in some instances go through direct germination as well. So similar sort of looking cycle when we take it as a diagram like that. But again, thinking about now adding the plant and the environment into that life cycle. So again, the asexual stages of the life cycle are exactly the same. And then you also have, in effect, another circle on the outside edge of this, looking at the sexual reproduction with direct and indirect germination as well. This part, when we were looking at Remorum, was aerial transmission. Now we're looking at soil and water transmission. So Synonyms we haven't found any signs of aerial lesions, and it's sort of very much soil-borne and through water courses as well. So as a result, when it enters into its host, those are going to be either within the roots or at the root collar at ground level. So if you're finding lesions on sweet chestnut that are in the air, it's most likely going to be Remorum. But if you're finding it at ground level, then it isn't Remorum, because as we've discussed, that's aerial transmission, then it's most likely going to be one of the soil-borne ones. And in this instance, we're looking at Synonyms potentially. So thinking about distribution, as you can see, it's exceptionally wide distribution over a number of different environmental types. Really likes a Mediterranean climate, 600 mil rainfall plus a year. So does quite well on the continent, in Spain and France, certainly southern France. But thinking about climate change and the types of environments that we're expecting to see here over the coming years, could well feature more in some of our talks in the future, looking at pathogens. Recorded, suspected that it was here late early 1900s. Difficult to say with any certainty, but looking at the records and the descriptions of what was found and observed at those times, it's been here for a significant period of time. Bringing it a little bit closer up to where we should be now, thinking about. So this map was 2017. So this was reports through to Forest Research for 2017. Again, thinking about the environmental types, very much a southeast focused, nice and warm, reasonable amount of rainfall. Not so much in the southwest. I'm not entirely sure why that is. Not a lot of sweet chestnut ground down there, but thinking about the wide host list, maybe maybe it's underreported down there. It's difficult to say really. So thinking about what symptoms look like on sweet chestnut. So again, we'll see the foliar collapse, as you can see from that picture on the left-hand side. But then looking at the stem infections as they start to come through, this picture here, you can see this sunken area protruding up from the ground. So if this area in the middle here, this will all be necrotic. The outside area where it's sort of normal diameter will be the nice green live material. So this is that sort of leading edge of that lesion coming up out of the soil. It's better to see the sort of contrast between the two here on this top right-hand picture. It's worked its way up through the stall, through the roots, and you can start to see it emerging out on the recoppiced stems as well as it's coming up through. That works its way up through. Again, sort of thinking about root-based stuff, you tend to get that foliar collapse a lot earlier on than you would with sort of infections up in the crown, because you'll get small bits and pieces that start to collapse in the crown if you have an aerial infection. But if the whole stem or a whole coppice stall starts to decline and foliar collapse like that quickly, it's likely to be a root issue, because it's just having that instant impact. Then as it starts to progress through, you'll end up seeing sort of bits of dead stalls and stems kicking around, to the point where it can become quite pronounced, where it's had a really good go through this one here, and it's had all the sweet chestnut out, and we're left with the sort of birch and ash and the oak bits and pieces kicking around the edge. as it's starting to work its way through to sort of the stage on the left, of the one on the right, you can see it's just restricted to sort of stress epicormic regrowth. This is the tree really sort of reacting and trying to just survive as best as it can at that point. But you know, it's not looking very good at all. So it can be quite a fast-acting pathogen. So it's not subject to any official controls with regards to statutory plant health notices. Again, sort of looking at distribution globally within GB, and also looking at sort of the host range and the period of time it's been here for. We're not going to affect any wide-scale control. It's more about sort of thinking about biosecurity with regards to equipment, as I was saying, sort of removing all that organic material and debris, and also plants for planting as well. And also sort of things like this, making people aware of what it is. I'm not quite sure what residents in this Western Australia area thought about this sign, as somebody's had a go at it with a shotgun, but this is warning of the Jarrah back in Western Australia. So, it has a significant issue over there as well. You can see sort of from the highlight from the sort of sky on the top there, a number of tops dying back within the Jarrah trees there, so causing them a serious problem. So we'll move on now to sweet chestnut blight. So the other two, thinking back to that phylogenetic tree to begin with, were heteroconts. There were mycetes in that Phytophthora genus. We're now looking at a fungal pathogen. So the other side of that tree primarily affects Castanea, sweet chestnut species, particularly North American sweet chestnut and European. Had a really significant impact in the States. It was found there a number of years ago and has caused death of millions of trees over there. Dentata seems to be far more susceptible to Cryphenetra parasitica than our European sweet chestnut does. But it has had an impact in Europe. They have been surveying for it over there for a number of years, and we have also since 2006. Our first finding in the environment in GB was in 2011 in a nut orchard in the Midlands. So looking at global distribution, again, you can see we've got a significant distribution across from east to west, really, across the globe, and down in Australia as well. So quite a dispersed pathogen, has been present within Europe for a number of years. You can see the sort of earliest introductions mid part of last century for a number of places. We were very much later into this century in 2011. But it's been about for a number of years, which has allowed sort of a fair bit of study of it on the continent. How does it get in to its host? It's a wound-based pathogen, so it needs cuts or grafts or some form of physical damage for it to get in. A number of movements have occurred in the past through grafted trees. That's one way it likes to move around. As I'm sort of looking beforehand, it can cause mortality. Oak can be a host sometimes. That's not been recorded in Great Britain. The places where it has been recorded, there's been an exceptionally high and significant spore pressure in the wider environment for that to occur. So basically, it's got to the point where it's been an overload of spore pressure rather than it sort of being a majorly susceptible host. So it does occasionally happen, but hasn't within Great Britain. The hypervirulence, so that's sort of what I was alluding to a little bit, sort of looking or talking about the research that's gone on the continent. It is possible to use a hypervirus to control Cryphenetra parasitica, but it has to be of the right vegetative the right mating type and the right I'm going to stumble over that word again, so I'm going to miss it. It needs to be compatible. That's the word I'm looking for. The main sort of symptoms that you see are the fissured bark, and you'll also see foliar collapse again, and these sort of numerous orange fruiting bodies, which we'll take a closer look at in a minute, and also my buff-coloured mycelial fans on the edges of those lesions as well. So again, sort of looking at life cycles and how it transports itself and transmits around and what it gets up to. So we'll start over on the right-hand side here. If my pointer, there it is. We'll come back up. So we'll start with the cankers. So the disease has elicited a reaction within the tree, and it will form these cankers. As they start to develop, it will then produce these little fruiting bodies, which I'll show you closer in a moment, as on the bark surface. Now, it has two ways of reproducing itself, through asexually, through these conidiophores. So they're sort of sticky tendrils that contain DNA that can then go on to infect another host. It will also, in some instances, if there are the right mating types present, reproduce sexually as well and produce ascospores. So they then float around in the environment, find a sweet chestnut tree to land on that's got a wound, work its way into the wound, and it starts all over again. So quite a reasonably simple diagram to show for you there. Thinking about symptoms as to what it looks like within sweet chestnut. So here we've got the sort of younger, small diameter material that we see. The lesions in the small stuff are very different from the larger diameter stem infection. So they're orange sort of coloured. Sometimes they're swollen cankers. Sometimes they're sunken cankers, like the picture on the right hand side. As you start to move up into sort of stem diameter and size, you get these really sort of shaggy, swollen lesions that are really quite pronounced from the rest of the bark. The patina of the bark, sort of either side of that tends to be quite smooth, as you would expect for chestnut, but where you've got an infection and a wound, funnily enough, at the centre of that one, you start to see this sort of swollen canker with platelets and fissures and all sorts going on. Sometimes you'll see from a distance they have slight orangey bands either side, and you'll also see epicormic growth around those as well. So you can see one there, this one in the foreground, and there was one in the background on this one somewhere as well, but I can't see quite now at the moment. Here again, sort of more pictures of cankers and what they look like. There can be quite a variation in chestnut bark. Sometimes it can have a sort of naturally sort of fissured appearance to it anyway, but again you'll see that the cankers are far more pronounced fissures than any that would be naturally occurring from sort of the normal morphology of the bark. And again, on the right hand side here, you can see a swollen canker sort of as it comes down towards this branch union here, and then you've got that canker sat in through the middle of it. So can present in a number of different ways, but those fissures are really quite one of the key things to look for. And like a lot of things that attack trees, the tree then tries to throw out a load of epicormic growths because it's under stress and not happy and trying to react to that infection as well. So with the cankers, you'll quite often see a proliferation of epicormic growth just above and just below those cankers as well. So it's something to keep an eye out for when you're looking through your stands. This pole stage stuff is nice and easy to assess for chestnut blight, because it really does give some good symptoms if it's present. As you can see, that one in the back there, nice big long canker, slightly orange at the bottom, and then you've got the epicormic growth coming out from underneath. Smaller stuff can, at lower levels of epicormic stuff, be a little bit easier to spot as well, because you'll get this flagging, this foliar collapse. So here, you can see just to the right hand side of the stem, you've got some chestnut leaves that aren't looking too good. And then you get a little bit closer in to have a closer look, and you can see the eagle-eyed, those of you will have spotted that little lesion down the bottom there, that orange coloured lesion. And then when you get a little bit closer, again, you can see sort of very typical of those lesions I was showing on the smaller diameter stuff at the start of this one. It's that nice sort of orange, bright orange colour. As that then advances through stands, you can then start to get sort of mortality of stands as well. So you'll see sort of the odd tree that's standing in there dead as well. You'll be able to tell if it isn't cinnamomi from this one, sort of again thinking back of sort of trying to diagnose between different stuff, because cinnamomi will have a basal lesion that's come out of the soil, whereas sweet chestnut blight won't. Thinking about sort of urban environments as well. This was a street tree in London we looked at a few years ago. You can see, I'll point out to begin with, a bit of street furniture that you can see here in the street light. So a bit of pruning had occurred over the years, as you would expect, to try and keep that street light free of foliage and stuff. So there was a number of wounds. There was an infection nearby in the woodland, and it's come out onto this one as well. We've managed to find a wound on this tree, and it started to cause dieback, as you can see from here, and in through this one as well. So yeah, a number of different sort of symptoms that you can look out for, but that sort of foliar collapsing and unexplained dieback is a really good one to sort of start to hone your eye in a little bit closer. Then when we're starting to look at the underbark symptoms, then we're looking for these mycelial fans. So again, thinking about fungus, it has to have mycelium. You can see here, this one a little bit dried up, but you can just see on the edge, little mycelial fans in underneath here, and a bit further back. This one's quite nice, quite a bit, well, it is fresh, it's just been opened up. You see the leading edge between the live healthy material and the dead material that the pathogen's been through, and you can just see some of the mycelial fans on the edges of that working through on that leading edge as well. When you get a bit closer and taking that panel out, they look a little bit like sort of moth wing prints almost to me. So you can see there's a number of fans in through here. And then looking closely underneath the hand lens, far more pronounced, and you can see that mycelial network stretching out on those leading edges. So that's where the disease is most active, and it's sort of a good place to take samples from when we're out surveying. So looking at fruiting bodies now. So we've got a tree here, we've got lots of epicormic growth going at the bottom, that's retained. It's the winter, as you can see, looking in the background, all the other broadleaf trees have dropped their leaves, but this one has still got significant amount of leaf on the epicormic shoots, which is a bit odd for sweet chestnut. Expect it for some oaks and beeches, but wouldn't think so for sweet chestnut. You can see there's a slight orange hue to the bark, and there's also significant wound up the top. When you get a little bit closer up to the side of the bark here, you can see it's got a sort of textured orange dimply effect going on. So these are all the fruiting bodies that I was talking about just now. They're starting to pop out through underneath the bark. And then when you take a hand lens, so this is a ×10 hand lens, you can start to see they've erupted out through the bark. So some things you'll get like coral spot, for instance, that sits on the surface of the bark almost. It doesn't look like it's erupted through, but this is really very distinct and different because you can see it's pushed through that bark, popped it open, and then appeared out through. So really quite pronounced through a hand lens and rather different looking. So another good diagnostic feature when you've started to zoom in to have a closer look. So sort of little quick recap on that one. So individual mating types can reproduce asexually, as we were talking about, but they can also reproduce sexually as well using ascospores. The conidia are waterborne, can also be moved around sort of in animal contact as well, but that's short distances. What I really wanted to show you with this slide is this little section here, again, sort of thinking using hand lenses. So again, a ×10 hand lens. So that triangular shape is that one there. And what you can see here is a conidial tendril. So you can see that conidia just appearing out of that fruiting body as well. So that's what they look like. You can actually see them with fairly low magnification. Quite fun looking little things, really. So thinking about distribution within England now, the map you can see on the left-hand side is probably familiar to most of you that have dealt with ash dieback and looking at the maps we produced for that over the years. We produce our maps on a 10K grid square basis, and the colours are the infected squares or squares where we found evidence of chestnut blight. So it is a quarantine organism, and as such, it's also like Remorum. There's statutory action associated with that in the shape of statutory plant health notices, and there's also sort of persisting biosecurity measures that surround those notices as well. The first finding was in 2011. Those were trees for nut production. There's been subsequent findings in 16, 17, 18. And then 20 as well. So those have been a mixture of sites. The ones down in the southwest were single tree infections, pretty much in plants for planting that hadn't spread any further. Some of the southeast sites are sort of on the wider environment type. So there's infections in woodlands rather than just single trees. Best way to report this is via tree alert again. And one of the best things that anybody can do with anything that I'm going to talk about today and tomorrow is sort of putting in place good biosecurity measures. That's one of the best things to break transmission cycles is thinking about your biosecurity protocols, how you're arranging your work, and also thinking about sort of the physical biosecurity of equipment and boots and things as well. So moving on finally to oriental chestnut gall wasp. So number of findings on the near continent over the past few years, from 2002 in Italy through to Switzerland in 2009. Finding here was in 2015. But this is what it looks like. It's a tiny little wasp, a couple of mil in diameter, in length, sorry. It reproduces parthenogenically, so there are no males, only ever females found. And it causes the larvae cause these reaction in the sweet chestnut buds, and it causes these galls that then develop on into all sorts of different shapes and sizes and colours. But those are the initial sort of rose-coloured crown of thorn type galls that people talk about sometimes. Looking at global distribution, as the name would suggest, oriental. There's plenty of it over in Asia, but also into Europe as well, and in North America. So thinking about life cycle, so we'll start with the galls, the nice fresh galls that appear as the leaves start to develop, those leaf buds start to develop, the larvae start to develop as well, and that elicits this response within the plant to encase it within this hard, well, to begin with soft, but then progressively hard gall. So that develops through the course of the season, changes colour to sort of a dark colour, dark greeny brown colour. The larvae will then pupate and emerge out through the emergence hole there. It will then find a suitable tree usually not that far away from where it's emerged from, and then the female will oviposit her egg into the leaf bud, which is what will develop into the leaf the next year. So she's laying her egg into that. It will hatch, go through probably, I think it's 1 or 2 instars, and then it overwinters and starts the process again. Like a lot of insects, it has a sort of peak emergence period as well. So you tend to get a lot of these that will emerge at the same time. It's a nice sort of survival strategy. So you'll get all those sort of female wasps arriving. I think the other year they were out sort of about June time. They need so many degrees to get to a certain level before they'll emerge. But now sort of thinking about the development of those galls, so they start off as the leaf bud starts to form in this really nice, easy to spot, bright sort of rose-coloured gall that's quite soft. They then start to develop as the leaf develops. Sometimes it will be a terminating gall, as you can see there, so you won't have any leaf at all. Sometimes there will be one that sort of appears in the midrib and you'll get a distortion. Other times they can be quite significant and you'll have a real distortion of the leaf and quite a large gall there as well. So they can, a number of different sort of strategies for forming those galls, but in effect they harden as the season goes on to protect them from predators. And then you might recognise this one from just now as a cut through from the other one. This is the emergence hole later on in the year. What will then tend to happen as the season goes on is those leaves they'll get to a certain size and then they'll die because they're not getting the nutrients that they need. So you'll see galls associated with these dead leaves as well sometimes. Sometimes you'll get these really sort of deformed galls as well, where you'll just get these tiny little leaf on the end, and then those can die off as well sometimes. And these are sort of some of the distortions you'll see as well. So it's really sort of a number of different things. But the good thing with this is it's the only thing, well, not the good thing from a diagnostic point of view is it's the only thing that will cause a gall on a sweet chestnut. So if you're getting galls on sweet chestnut, it's oriental chestnut gall wasp. A little bit more difficult to spot up in the canopy. So that's what you tend to be looking for, is that sort of cluster of dying leaves. And you can get your binoculars out and sometimes you'll see distortion. Sometimes you won't if they're too high up in the crown. But another useful diagnostic thing with these is that the leaves with galls tend to persist on into the winter. So here you can see with these two pictures, these were taken in tail end of November, sorry. So you can see they've persisted through from the summer all the way through, and you still can see those galls that are there. So significance for gall wasp, thinking with chestnut in itself, it's not going to kill the tree, but what it will do is it will cause a general decline, or likely cause a general decline in vigor, especially if that tack is heavy and sustained over a period of years, because you're losing the available photosynthesis area in effect, so you're reducing its ability to produce energy and food. So by itself, not bad. Combined with other things, it's not so good because it's weakening that tree and making it more susceptible to other pests and pathogens. So looking at distribution, this map is a little bit out of date. This was from just about this time last year, but as you can see, there's a number of findings. London down into sort of Hampshire, across into sort of working its way along the M4 corridor as well, down into Kent. I think there's probably a little bit more out there than this map suggests because it's quite difficult to survey for it unless you actually look actively searching for it in a small area within a woodland. It can be tricky to pick up on it sometimes, but it is reportable again through tree alert. One of the interesting things that's been observed in the southeast, looking at sort of native controls, is there is with oak galls, there is a native parasitoid wasp that targets oak galls. There's also been found to be targeting to a certain extent oriental chestnut gall wasp galls as well, so that's useful. There is a biocontrol agent, another parasitoid wasp that's present within the gall wasp's native range, Tormus sinensis it's called. So there's they use that over on the continent, and there's also currently a period we're looking at using that here in the UK at the moment, but there's also a period where we're looking. Sorry, I'm stumbling on my words again. Apologies. We're looking to use it here through legislation, and we're just trying out that at the moment through consultation. That's the word I was trying to find. Right, and that is my last slide, and I shall now stop bumbling and have a drink whilst Caroline tells me what questions I need to answer.
Thanks for that, Alan. That was brilliant, really concise. So at the moment, we've only got 3 questions. So I'll start off with one from Ron, which is Remorum temperature sensitive?
Yes, it likes the temperature range running through from top end of about 24 down to about lower end, sort of, it starts to become fairly inactive at around about sort of 10 degrees. So the colder it gets, the less it likes it. So it definitely has an ideal temperature range, definitely, but also humidity as well. It needs that certain level of moisture for it to be a suitable environment as well.
Lovely, thank you. Next question is from Angela. Can Remorum infection look like other diseases on rhododendron? Can it be confused with something else?
There are, yeah. So there's a number of other fungal pathogens that will have a go at rhodi leaves. One of them, or a number of them, they're grouped together because it's very difficult to tell the difference between them visually, but they cause a leaf spot. But there's a good way of diagnosing that, because if you hold that leaf blade up to the light, you'll see a crimson halo around that spot. So that's definitely not Remorum. So that's one of them that you'll come across quite often, which is easy to rule out. There isn't much else on rhodi that looks like Remorum.
Lovely. And our final question from Arno is, can cinnamomi symptoms be confused with parasitica? I've seen similarities for both in the Liphook Milland area on the boundary between Hampshire and West Sussex. So can you confuse the two?
I suppose from a distance possibly, but you won't get the fruiting bodies of parasitica that you will do with cinnamomi. So that's one good way to distinguish the two. And also thinking, as I was talking about cinnamomi, it's root based, so it's going to be starting at the ground. So unless you've got a cut stump that's infected with chestnut blight that's starting to sort of kill the fresh growth off, then it would be difficult to confuse the two. It's very much looking at you'll find the fruiting bodies with chestnut blight, and you'll find that soil-based lesion that will start with cinnamomi. So if you can sort of think about it and split it out into those two, that will help you out trying to sort of distinguish the two. And if you're not sure, ultimately put it through tree alert to report it.
Brilliant, thank you. We have no other questions in the chat, so unless there's anything on the floor, if anyone wants to use the raised hand function and ask their question. Oh, got a follow-up question from Arno. Are the fruiting bodies present all year?
So that would be for chestnut blight. If they're there, yes, you can see them. They might not be active, but you can certainly see the old ones from previous years as well. If there's a few there, yeah, you can definitely see them.
Okay, another one has come through from James. I see a lot of bleeding lesions on oak in Surrey, West Sussex area. Could this be Remorum, oak decline syndromes and or armillaria? And is there a quick field test for any of this?
So if you're seeing a lot of bleeding oaks, I would suggest that unless you've got an absolute shed load of infected larch or rhododendron literally right next to that oak tree, it's exceptionally unlikely, because it's a terminal host. So you don't, our native oak, the quercus are nowhere near as susceptible as the tan oaks in America, which is where the name sudden oak death comes from. So I would suggest it's not Remorum. It could well be armillaria, definitely, because that can cause stem bleeds. Easy way to distinguish with that is you'll see an awful lot of white mycelial activity underneath the bark. If there's loose bits of bark you can peel away. You'll also see the rhizomorphs, which is sort of what known as the bootlace, the licorice bootlaces. Or if it doesn't look fungal, then it could well be bacterial as well. So there are a number of bacteria like having a go at oak. So it could well be acute oak decline or chronic oak decline.
Okay, Andrea Griffiths, you had your hand raised briefly. I don't know if you're happy or if you want to unmute yourself and ask a question.
Yeah, sorry, I couldn't type for some reason. Just a quick question. We've got a lot of volunteers that often work in woodlands, etc. Obviously, you know, outside of COVID restrictions, obviously. I just wondered, what's the best way to make sure we're ticking a biosecurity box? Is there a sort of one product covers all spray that we can give them to spray on their boots, or what would you advise in general about biosecurity?
Well, I think it sort of links into one of the other talks later on in the week, but I'll give you a quick potted answer for that one. It's trying to take I'll try to use this word liberally, holistic view of biosecurity. It's not just about, you know, spraying some disinfectant on boots. It's about how you plan your work. So thinking about if the volunteers are going to be near a high risk area, maybe they did that at the end of the day rather than the beginning of the day. It's about sort of, you know, moving from one woodland to another, removing mud and soil from tools and boots. There is a disinfectant that we use called clean kill sanitizing spray. So that works on a number of fungal and oomycete pathogens. So you can use that as well. But yeah, it's about ultimately the real trick of it is about planning your work sensibly and smartly and reducing that risk and thinking about all of those risk areas and pathways as well, sort of with plants for planting and sort of thinking about where you buy those from, what questions you're asking of your suppliers. Maybe if you have the ability to do so, maybe quarantining those plants in your own facility for a little bit to make sure that there's nothing that's come in with them. But I'm sure Rob will give, if you know, it's a nice plug. If you sign up for Thursday's event, I'm sure there'll be some more information there for that one.
Thank you.
Yeah, that's brilliant. Also, if you want to Google the Forestry Commission's Keep It Clean campaign, there'll be more information on our website as well. We've got a question from Emma. What do we do if we identify cinnamomi? What's the best course of action in a sweet chestnut coppice? Again, it's coming back to biosecurity really. If it's there, there's not a lot you can't remove it from the site. You're not going to get rid of it and eradicate it from that site. So you have to think about planning your work and how you're going to do that and being careful about the timing of operations. So thinking about not doing it in wet and damp days where you might start to transfer it from, you know, one stall to another. It's about thinking about your biosecurity and being quite rigorous. If you know you've got something like that there, I mean, you should be doing, I think personally as sort of a professional, we should be doing biosecurity when we move from one site to another. But you want to be even more rigid about your biosecurity approach when you know there's something like that on site, because the risk of transmitting it from one site to another is quite high. So yeah, plan your work, again thinking smart. If you've got that infected site, do the work there when you're due to do maintenance on the equipment that you're using on that site, for instance. So you're not doubling up on the operations you need to do for that biosecurity. But yeah, I'm afraid if it's in there, there's not a lot you can do to stop it, I'm afraid. Okay, final question, I think, unless anyone has anything else, is from Julian saying, is there anything similar impacting silver birch? As we've seen a lot of lesions on those as well. And also alder is really suffering in the Hoe Valley in Woking.
A couple of things there. The alder, definitely. So that'll be another Phytophthora olnei. That's found in pretty much every watershed within GB now, apart from some of the really remote upland ones, very effective pathogen for older, and you'll see that dieback in decline and those spotty sort of sooty lesions on the bark. So again, it's waterborne. So once it's into a watershed, there's not a lot you can do stopping it spreading downstream. So if it's there, in effect, that's something you just have to live with and mitigate the risk that those trees are presenting to you from more from a health and safety management point of view, really. The other question, birch and lesions. Yes, I've noticed some lesions on birch over the past few years. I'm not sure whether that's because I'm looking at them more often than not. The ones that I've come across have been bacterial. The swabs that I've taken, I'm just trying for the life of me remember what they were, and I can't remember what they came back as. But it wasn't an unusual find, and perhaps maybe thinking a little bit more about the sort of drier summers, the hot, sharp, dry summers that we've had. Maybe some of that has been caused by stress that those trees are experiencing that they haven't done before as well. So leaving them a little bit more susceptible to other things.
Fantastic. Well, there's appears to be no other questions, no other raised hands. So just want to say really big thank you, Alan, for your presentation today. Thank you to everyone who's attended. Like I say, the recording of this will be going up on our FC YouTube channel. So do keep an eye out for that. And yeah, I'll be sending the questionnaire out on Friday. So if you can give us a response to that and let us know how you thought this went, that would be fantastic. But apart from that, everyone, I hope you all have a fantastic day. And yeah, hopefully see some of the rest of you on the other seminars this week.